All right, so we were talking about nervous tissue in chapter 12, and we're going to go a little bit deeper now. I'm going to be using this multipolar neuron as our example, but we need to start talking about what we call the transmembrane potentials. And there's a transmembrane potential means that there's a charge on the inside of the cell versus the outside of the cell. So it means the charge in the intracellular fluid is going to be a different charge than what's in the extracellular fluid. Okay? So we need to talk about how the cell, when it's at rest, it has, and these are transmembrane potentials. Okay, and there's three different transmembrane potentials we're going to look at. The first one we call the resting membrane potential. And the resting membrane potential, we're just looking at this cell when it's undisturbed, nothing's touched it, nothing's stimulating it, and what is the charge inside the cell body? So what's the charge inside there? At rest. That's the resting membrane potential. So we have to talk about how we even get to that resting membrane potential. The resting membrane potential in the neuron that we're going to be talking about is negative 70 millivolts. Okay, so the inside of the cell is going to be negative 70 millivolts. And then if something disturbs that cell and positive charges come into the cell, now we're no longer at rest, and we're going to be, that cell then is going to become more positive, and we call that a graded potential. And the graded potential is, they happen at that axon hillock. And they don't move any farther than that. The axon hillock kind of funnels all the positive charges into this one area. The positive charges are going to raise that resting membrane potential up to a negative 69, all the way up to a negative 61 millivolts. So that's our graded potential. And if the potential gets even higher than that, if it gets even more positive than that, and it hits a negative 60, then we will have an action potential. And the action potential starts at negative 60, which we call threshold. And it goes all the way up to a positive 30 millivolts. And an action potential starts on just a small piece of the axon. So it'll be on this initial segment of the axon to begin with. And then it'll go, and then we'll have another action potential on the next segment. And then another action potential on the next segment. And we'll have action potentials that just keep occurring all the way down that axon. Okay, so you get the point. They just keep occurring all the way down. We get action potential after action potential. Finally, at the end, those action potentials will get to that synaptic terminal, and we'll talk about what happens at the synaptic terminal. We've already seen what happens at the synaptic terminal at the neuromuscular junction. So there's a lot going on right there. And we have to look at how, first of all, we have to look at that resting membrane potential. How do we even get to negative 70 millivolts? What is causing the inside of the cell to be more negative than the outside of the cell? Okay? All right, so let's look at oh boy. Okay, let's look at this picture here. This is the neuron. This is the cell body. Whoop. So here's our cell body. These things right here, those are the dendrites. And then this thing over here, this is the axon. Okay. So when we talk about the resting, this is the resting membrane potential. And we said that the resting membrane potential is a negative 70 millivolts. There's a lot of things on the inside of the cell. So this negative 70 millivolts, we're really just talking about what happens just on the inside of the membrane. So the inside of the membrane all the way around here, that's going to be negative 70 millivolts, because on the, further in, you've got the nucleus and you've got all these organelles, and so we're really just talking about what happens on the just inside that cell membrane on the neuron. Okay. Well, the first thing that we have inside this cell, which is the easiest thing to tell you, is that there are these negatively charged proteins. So they have a negative charge to them. They carry a negative charge. So they're going to bring the charge, like a neutral charge would be 0. So since there's negatively charged proteins, they're bringing that negative, they're bringing the charge down to a negative number, right? But then there's two really important ions that we talk about. We have the sodium ions, and we have potassium ions. Okay? And we already know a couple of things about sodium and potassium. We know that sodium is higher concentration where? Outside the cell. So sodium has a higher concentration outside the cell. And based on our law of diffusion, that means sodium wants to move which direction? It wants to get inside the cell, okay? So all over these dendrites and all over the cell body, we have these leak channels, these sodium leak channels. And they're all over. And leak channels mean, do you guys know what? They're leaky. They are always, whoops, wrong, not there. They're always open. These sodium leak channels are always open, right? So sodium wants to move which direction? Sodium wants to move into the cell. It's carrying its positive charges into the cell. And that's kind of counterproductive to what we said, right? We said we want the inside of the membrane to be negatively charged. And now we have sodium ions that are leaking in and bringing in positive charges. Right? But we have these other leak channels, and these other leak channels are sodium or potassium leak channels. So now we have potassium leak channels. Okay. These potassium leak channels are all over the dendrites and the membranes as well. So they have many leak channels on the cell membrane and on the dendrites. So potassium wants to leak too. And what did we say about potassium? Potassium was going to diffuse which direction? Out. Potassium wants to diffuse out of the cell because there's a higher concentration inside the cell than outside the cell. Right? So if the same amount of potassium were moving out, which carries a positive charge, as sodium moving in, which carries a positive charge, those charges would just, yeah, they would cancel each other out, right? But there's more potassium channels than there are sodium channels. So we say there is a higher potassium permeability. Right, so let's talk about that, think about that. Potassium has a positive charge. As potassium moves out of the cell, what is it doing to the charge inside the cell? It's making it more negative because it's carrying a positive charge out. There's more potassium channels, leak channels, than there are sodium channels. Okay? If there weren't any sodium channels and it was just potassium leaking out, it would go all the way down to a -90 before it stopped. Okay? But because we have some sodium moving in through those sodium channels, it's not going to go that low. It'll go as low as -70. So when the question comes up on the exam, what creates the the resting membrane potential? Number one, the easy one is there's negatively charged proteins. And then number two, you'd have to talk about sodium and potassium, how sodium moves into the cell, potassium moves out of the cell. There's more potassium channels than sodium channels, and that allows that charge inside the cell to get to a -70 millivolts. So that's the very first thing we have to learn. You have to learn what causes that. Okay? Are there any questions on that? You have to know what causes the resting membrane potential. Now, the resting membrane potential is going to get out of sorts, once in a while. If there's a stimulus, it's going to move. It's going to move from a -70. It could get lower. Most of the time, it's going to get higher because as we talk about depolarization, it's going to get higher as it gets stimulated, but sometimes it can be inhibited and it can drop lower. So we'll talk about those things. But whenever it does get stimulated and it moves away from -70, we have a couple of things that are going to bring it back to -70. So first of all, those leak channels will bring it back, help to bring it back. They're always trying to bring it back. But the other thing is, as potassium is moving out, we're losing a lot of potassium inside the cell. And a lot of sodium's moving in, so we're accumulating a lot of sodium inside the cell. So we also have this pump, if you remember this pump, that's called the sodium-potassium exchange pump. Right? So we're losing a lot of potassium. So that exchange pump is going to pump potassium back in. And we're accumulating too much sodium. So sodium is going to pump, this pump is going to pump sodium back out. 3 sodium to every 2 potassium. 3 sodium will be pumped out to every 2 potassium being pumped in. That's the exchange pump, and that's how we will keep that cell at the normal concentrations of sodium and potassium, and to keep that transmembrane potential, that resting membrane potential, at a -70. All right? Okay, so that's our first transmembrane potential that we need to talk about. Then the second one that we're going to talk about is the graded potential. here. Okay, a graded potential. So a graded potential is when the cell is stimulated and it's going to move away from that negative 70 millivolts, right? So there's another type of channel that's on the cell membrane and the dendrites, and it's not a leak channel. These channels are called gated channels. So whenever we talk about channels, we're always calling them, what ion they are. So they're going to be either sodium channels or potassium channels. That tells us what ions move through it. And then we'll also give a description as to, are they leak channels, which are always open and ions can just freely move across, or are these gated channels? And the gated channels mean that they're closed until they're stimulated to open. So some of these channels are going to be stimulated to open if a chemical binds to it. And if a chemical binds to it, then we call it a chemically gated sodium channel or potassium channel. Chemically gated sodium or potassium channel. And these are the ones that are going to be involved in the graded potential. There are some other types of gated potentials on the dendrites and the cell membrane, like mechanical and voltage, but chemical are the most common, so I'm going to stick with that. It'll be less confusing, right? So these channels, and we're looking at this cell and we're saying, okay, the cell is at negative 70 millivolts. We know why it's at negative 70 millivolts. We're moving past that. Now we're saying there's a stimulus. And so we'll talk about a chemical that we've already talked about, which is called ACH, acetylcholine. We talked about acetylcholine binding to the muscle, the motor end plate, but now we're going to talk about acetylcholine binding to this neuron. Okay? So there are these acetylcholine receptors on these gated channels. Let's talk about that. All right, so on the dendrites and on the cell membrane, you have these channels. Now these channels are gated channels. Okay, they're gated channels. I've just kind of put them randomly around there. And they are chemically gated. That means that they're going to have a receptor on there that will bind to a chemical. And our chemical is acetylcholine. We're going to use acetylcholine as the example. So on each one of these receptors, or on each one of these gated channels, we have a little area on them that will bind to acetylcholine. So that's a receptor on the channel, the gated channel, and acetylcholine can bind to that. So the gates are closed until acetylcholine, and we'll make acetylcholine, I'll make it green here. When acetylcholine binds to the receptor on these gated channels, the gates will open. Okay. If acetylcholine is not bound to those receptors on those gates, the gates won't open. So acetylcholine is kind of like the key that opens up the gate. Now, if those gated channels are sodium channels, that means only sodium can come in through the gate. If they're potassium channels, then potassium could leave through the gate. But we'll start by saying we're going to talk about these sodium channels, and we're not going to talk here about the potassium channels. We're only going to talk about, so we'll just cross that out. We're only going to talk about the chemically gated sodium channels. When you get to advanced, we'll talk more about the potassium ones. All right, so there's some type of stimulus, and acetylcholine was released. Acetylcholine binds to the receptors, and sodium, which is higher out here in the extracellular fluid, sodium moves into the cell. So this is in addition to those leak channels. Those leak channels are still open. They're still letting sodium in, but that's just keeping it at -70. Now we have additional channels being opened. These chemically gated sodium channels. Something caused acetylcholine to be released out into the extracellular fluid, and now acetylcholine has bound to that receptor. Acetylcholine has bound to the chemically gated sodium channel. Sodium starts to come into the cell. What's going to happen to our resting membrane potential as sodium comes in? Sodium has a positive charge. It's going to get less negative or more positive, right? So what is more positive than -70? -69, right? And then more sodium comes in. The more acetylcholine there is, the more gates open up. So then the charge, more positive charges are coming in. So then the charge moves to -68 and then to -67. It'll keep going positive as long as acetylcholine is in that key, keeping that gate open. It'll go as high as -61. And all those positive charges that are coming in, they're kind of pushed to the outside. They're kind of pushed right to the edge of the cell membrane. And there's a current inside the cell with all the cytosol. And the current pushes all those positive charges down here to this axon hillock. So they're flowing by a current, and they're starting to accumulate right in here at the axon hillock. And it'll go as high as -61, and then it could just fizzle out. So if acetylcholine stops being released, or not enough acetylcholine was released to open enough gates, the charge might only go to -61. So we might have a charge that went from -70 and then finally up to -69 to -61. This here, the -69 to -61, that's what we call a graded potential. On the neuron, that doesn't, it for our sake, for this purpose, it stops right there. And it's not going to go anywhere else on that neuron. And it fizzles out. And so we call it a graded potential. So a graded potential, we could say, is local, because it only goes as far as that axon hillock. It's not going to go any farther. And we can also say that it's temporary. It's local and it's temporary. It's not going to create anything else because there's not enough acetylcholine, there's not enough gates open, there's not enough positive charges moving in. So it's local and it's temporary. Okay? Now in other cells in your body, a graded potential could, like if it were a secretory cell, if it were a mucus cell, or a cell that secretes saliva or sweat, a glandular cell, that graded potential alone could cause that cell to secrete. But on the neuron, it will, it could facilitate it, which, we'll talk about in advanced, but unless it goes higher than -61, it fizzles out. It just fizzles out. Okay. So let's talk about then, so that's the graded potential. The graded potential is local, it's temporary, it's due to these chemically gated channels on the dendrites and the cell body. It only goes as positive as a -61, and then it fizzles out. So that's what a graded potential is, right? Now we're going to look at the next type of transmembrane potential, which is called an action potential. And an action potential is if there's enough positive charges moving into the cell that it hits negative 60. So funneling down into this axon hillock, if there's enough positive charges that raise that axon hillock to a negative 60, now we've hit what we call threshold. And once you hit threshold, an action potential will occur no matter what. It's called the all or none principle. You hit negative 60 and that neuron is going to create an action potential, will generate an action potential. Okay, so to make this clean, I'm going to go to this slide with a blank. All right, so we're talking about an action potential. And so as a little recap here, we went from a resting membrane potential of negative 70 millivolts, and then positive charges, sodium moved in. All those positive charges funneled down to the axon hillock, and the charge went up to a negative 61. That's a graded potential. And now at this initial segment of the axon, we actually call it that. We call it the initial segment. It's the very first segment of the axon. It's right after that axon hillock. Okay? So there's enough positive charges funneling down towards that axon hillock and towards that initial segment that the charge went to as positive as negative 60, and that is our threshold. So now we have another gate. We have another gated channel that we're going to talk about. And these channels are only on the axon hillock. Okay? These gated channels that we're going to talk about are they are voltage gated sodium channels. So this tells us that what ion can move through it? Sodium. And what is it going to be sensitive to? Voltage. Okay, so they're not chemically gated anymore. Now they're voltage gated. So they're going to be sensitive to voltage changes. And our very first voltage change is a negative 60. So those sodium gated, the voltage gated sodium channels are sensitive to negative 60. Okay. So we'll draw a couple of those channels here. There's many, hundreds of them. Many sodium channels. These voltage-gated sodium channels are going to be sensitive to a couple of different voltages. They're sensitive to negative 60 millivolts, and they're sensitive to positive 30. Okay? And then they're going to be sensitive to negative 70 again as it comes back down. Right? As the charge comes back down. So let's look at that. So the charge hits at the initial segment, it gets as positive as negative 60, and that negative 60 charge causes all of those voltage-gated channels on that initial segment to open up. So all the sodium channels are opening up. Sodium then is going to flood into that axon hillock. Floods into that axon hillock. Positive charges flooding in. So our negative 60 is going to get even higher. And what's higher than negative 60? negative 50, negative 40, negative 30, negative 20, negative 10, 0, positive 10, positive 20, positive 30. It's going to get all the way up to a positive 30. And at positive 30, the sodium channels slam shut. So if they slam shut, if they shut at positive 30, that means no more sodium can come in, and that means that charge cannot go any higher than positive 30, right? But then we have some other voltage-gated channels, and those voltage-gated channels are potassium. So now we have voltage-gated potassium channels. They're also all over the axon. So these are all over the axon. And the voltage-gated potassium channels are sensitive to voltage too. Tells us voltage-gated, meaning they're sensitive to voltage. Potassium channels, meaning they're only going to allow potassium to cross. So the voltage-gated channels are sensitive to positive 30 millivolts. Okay. And then to the negative 70 millivolts. So they're sensitive to those things. So as those sodium channels close, the charge inside the cell reached positive 30 millivolts because all that sodium was flooding in, and then sodium channels closed, right? Then the potassium channels open, and potassium does what? Exits the cell. So potassium always exits the cell. Unless it's being pumped back in, potassium will always exit the cell. So now we have positive charges leaving. So now what's going to happen to the inside of our cell? It's going to become more negative again. Positive charges are leaving, it becomes more negative. It was at positive 30, so it goes down to positive 20, positive 10, 0, negative 10, negative 20, negative 30, negative 40, negative 50, negative 60, negative 70. So it moves the charge inside the cell from positive 30 all the way down to a negative 70, right? And at negative 70, the potassium channels start to close. And I say start to close because they don't all close at the same time. Some are closing, others are still open, so the charge will dip even lower. It'll go as low as a negative 90. And at that point then, all the potassium charges will close. So at negative 70, some close, and at negative 90, all close. Okay? And then the action potential is over. But now we've got to bring it back up to resting. And what did we say brings it back up to resting? Leak channels and sodium-potassium exchange pump. They will bring it back up. And so now our action potential is over. We're going to go through this chart and then we'll take a break. Okay. This is a graph hopefully will help you a little bit. So I'm going to highlight this as I go along. I'll highlight in yellow. So this is when the cell here at negative 70, it's sitting at a resting membrane potential. Leak channels are always open, sodium's always moving in, potassium's always moving out, the sodium pump is going, sodium-potassium exchange pump is going, and it's keeping that cell at negative 70 millivolts. Then we hit this point right there, and we get a stimulus. Okay, so the stimulus is acetylcholine binds to the chemical gated channels, sodium channels, right? So acetylcholine is binding to the chemical gated sodium channels, and where did we say those were? So these are on the dendrites in the cell body. So acetylcholine binds, and the sodium channels, these gated channels open up, and sodium moves into the cell, and then we said the charge does what as positive charges move in? It becomes more positive. And so here we start to see the charge becoming more positive, and we're at negative 69, and we go all the way up to negative 61. And what did we call that when we go from negative 69 up to negative 61?
Graded potential.
That's right. So that right there is our graded potential. So the graded potential is occurring at the axon hillock. All those positive charges are funneling into that axon hillock. The charge gets as high as a negative 61. So from here to here, that's our graded potential. It's local, temporary. It's not going to go anywhere unless it hits negative 60. Okay? And then we have another point right here, and that's that graded potential has now hit negative 60. The graded potential reaches threshold. So now we've hit threshold right here at negative 60. That's our threshold. What happens at negative 60 on the initial segment of the axon?
Voltage gated channels open.
Right, so here we have voltage gated sodium channels open. And sodium starts flooding into that initial segment. This is at the initial segment. Sodium's flooding into the axon, flooding into the axon. So our charge is going to become more positive, which is what happens. We see this positive, the positive movement on that graph, right? Whenever we have positive movement, we call that depolarization. So that's a name we give. Depolarization. And because this is happening so rapidly after that the voltage-gated charge is open, we'll even call it rapid depolarization because it's happening so quickly. So sodium is flooding into the cell, and our positive charge shoots all the way up to a positive 30, right? The cell's getting more and more positive. And what did we say happens to the sodium channels at positive 30? So the sodium channels close. And what did we say happens to the potassium channels at that point? The potassium, so these are voltage-gated potassium channels because they were sensitive to voltage, they're sensitive to positive 30, and they open up. Potassium always does what? Unless it's pumped, it's always going to move in what direction? out. So potassium moves out of the cell, taking its positive charge with it. So now the charge inside the cell drops rapidly. And when it drops like that, we call that repolarization. Repolarization means that we're bringing it back to rest. We're trying to bring it back to rest. Depolarization means action. Repolarization means we're bringing it back to rest. So those the charges inside that initial segment drop all the way down. They're going to drop down to about -60, -70. And at -70, that's when the potassium channels will start to close. They start to close. Not all of them close, though. So more negative charges, more positive charges move out, and it drops the charge inside that initial segment will drop all the way down to -80, -90. And then at that point, all the potassium channels are closed. And to bring it back up to rest, it is the leak channels and the sodium-potassium pump that brings it back up to resting. Everything else that you are going to learn is going to it's going to reflect on this. When you get to advanced, you're going to have to go over this again and go much deeper into this. When you take a pharmacology class and you're talking about drugs, it all comes back to this with the nervous system, if there's drugs that affect the nervous system. And drugs, any ionic drug is going to affect this. And so this is the basis of it all. You have to learn this. So you have to tell the story, right? So you have to explain in painstaking detail what happens at each spot, you know? You have to know, starting from the resting membrane potential. This is how we get to the resting membrane potential. Then to a graded potential. This is how the cell starts to become more positive. And then the action potential. This is how an action potential occurs. And an action potential, we're going to learn when we come back from break, is going to propagate all the way down that axon. It doesn't just stop at the axon, the initial segment of the axon. It's going to go to the second segment, then the third segment, then the fourth segment, all the way down to the end of that axon. So this is something you have to know. And once you know it, it's a story. It's easy to tell it once you understand it. And that's what I want you to do. I want you to be able to tell this to somebody else. So, all right, we're going to take a break. All right, so I want to explain to you this principle that we call the all or none principle. So again, when we're looking at this chart, when you're at -70, -70 here, that is your resting membrane potential. When the charges in the cell go from -69 to -61, that is your graded potential. So it's those gates, those are important for you to know. There's going to be questions on that. Where are the leak channels found? Well, they're on the dendrites in the cell body. Where are the chemically gated channels found? Well, they're on the cell membrane and the dendrites. Where are the voltage gated channels? Well, they're on the axon, starting from the initial segment on down, all right? So it's important to know those, and it's important to know what's happening with the ions. Are they moving in? Are they moving out? What's happening with the ions? Now, when we finally hit threshold, and threshold is negative 60 millivolts, that's when our all or none principle kicks in. All or none. Which means you either hit 60 and negative 60 and you get an action potential, or you don't hit negative 60 and an action potential will not occur. It's one or the other. You're hitting threshold and you get an action potential, or you're not hitting threshold and you only have a graded potential. Okay? One or the other. That's what the all or none principle states. Okay? Doesn't matter how large the stimulus is. It doesn't mean if you have massive amounts of acetylcholine that opened up those sodium gates are only enough to get you to threshold. No matter what, you are going to get an action potential if it hits negative 60. Okay? Okay. Then the next thing is we have this thing called the refractory period. And the refractory period means that it's a period of time during this action potential when you can't have another action potential at that same segment. It prevents one action potential from occurring right on top of the other action potential at the same segment. You have to complete this far enough, this action potential far enough before another action potential can occur. All right, so we hit threshold and the sodium gated channels, the voltage gated sodium channels open up, and so we're getting depolarization. Another action potential cannot occur right there. Why? Because the sodium channels are already open. You're not going to open anymore. It's only going to go up as high as positive 30. You're not going to get another action potential right there. Not going to happen. Then the sodium channels close and potassium channels open. Potassium starts to move out of the cell and you start to repolarize. At this point, the sodium channels are inactivated. They can't open. So all along this repolarization, sodium channels can't open. They are inactivated. Okay? They're inactivated. They cannot open. So we can't get another action potential. We have to have those voltage-gated sodium channels to open in order to get another action potential. So we're not getting that. And then finally, when the negative charge gets down to a negative 70, that's when the sodium gates regain their resting potential, their resting potential. That's when they can open again. Until then, they can't open. So we call that the refractory period, right? Where another action potential can't occur. I'm not going to go distinguish between absolute and relative right now. So just know that the refractory period means another action potential cannot occur during those times. During rapid depolarization and repolarization, another action potential cannot occur. Okay, a couple more concepts here, then we'll be done. So this here is showing the axon. This is the initial segment right here. So we talked about the action potential how it shot up to positive 30 and then back down to negative 70, then back down to negative 90, then back up. So we talked about the whole action potential. That action potential, as those positive charges are moving in from the sodium, there's a current inside that axon. There's fluid in there, that intracellular fluid, and that has a current that's going to carry those positive charges down to the next segment. And it will cause that next segment then to hit threshold. And then that next segment will have an action potential. And sodium ions are going to rush in, and those positive charges are going to move down to the next segment, the third segment. And that segment's going to reach threshold, and we're going to get another action potential there. And that's just going to keep happening. Sodium ions are going to move in, the positive charges move down to the next segment, and you're going to get another action potential there. And that's just going to keep happening all the way down that axon. And that traveling of the action potentials, when we generate a new one, the next segment down, and then a new one, the next segment down, and then a new one, the next segment down, we call that propagation. Propagation. That means that action potentials are being generated all the way down that axon. And all those action potentials together, we call a nerve impulse. Nerve impulse, right? So a nerve impulse travels all the way down to the synaptic terminal. Now, in an axon that we're seeing here that doesn't have myelin around it, every single segment of that axon has to depolarize, meaning every single segment has to have an action potential in order for that action potential to travel all the way down to the other end of the cell, to the other end of the axon. So we call that, when it doesn't have any myelin on it, we call that continuous propagation, because the initial segment had to depolarize. We had to get an action potential there. Then the second segment had to depolarize, and we got another action potential there. Then the third segment depolarizes, and we get an action potential there. Then the fourth segment depolarizes, and so on, until we get all the way down to the other end. Every segment has to depolarize. But when we look at a neuron that has that's myelinated, and if you remember what myelin is that waxy lipid that covers the axon. And so it's covering over the top of the axon. So we're looking inside the axon here, and you can see that there's positive sodium charges there's positive charges inside the cell. And we can see sodium outside the cell here. Well, sodium can't get through that lipid, that waxy myelin. So the only place that sodium will actually be able to enter the cell through those voltage-gated sodium channels is wherever we have a node. So where we have the myelin, that's called an internode. And where we have a bare segment of the axon, that's called the node. So that's really the only place that sodium can enter. So that node is the only place, the only segment on that axon where we're going to get an action potential. So we get an action potential at the initial segment, and those positive charges from sodium are going to get carried down to the next node, and then we'll get an action potential at that second segment, and as sodium comes in, those positive charges move down to the next node, and then that opens the voltage-gated sodium channels at the third node. Sodium comes in, and the positive charges move down to the fourth node, and we get another action potential there. So that's how action potentials travel when you have a myelinated axon. You can only get an action potential wherever the axon is bare at those nodes. So it jumps. Those action potentials are kind of jumping from the initial segment to then this segment to then this segment So it's jumping, right? So which one do you think would be faster? And this one, by the way, this is called saltatory. Saltatory. Saltatory propagation. Do you think that continuous propagation or saltatory propagation would be faster? So saltatory is going to be faster. In saltatory propagation, you do not have to depolarize this segment or this segment or this segment. You're skipping segments. So you are passing that action potential much faster because you're skipping those segments. You don't have to, in this segment here, you don't have to go all the way up to positive 30, then back down to negative 70, then back up to, down to negative 90, then back up to negative 70. You don't have to do that. You're skipping it. So it's like skipping in line. How are you going to get to the front of the line faster? If you skip people in line, you get to the front faster. Well, that's what happens in saltatory propagation. The action potentials are skipping segments to get to the front of the line. Right? Okay, so saltatory, so on your myelinated axons, that's why the majority of your nerves, the neurons in your body, are going to be myelinated because they can send messages a lot faster. There are some that are unmyelinated. Most are myelinated. There's also some in the peripheral, there's some in the brain, some in the peripheral nervous system, in the autonomic nervous system that are unmyelinated. But there are diseases that cause demyelination, like multiple sclerosis. Multiple sclerosis takes off this myelin on these axons that are normally myelinated. And that's going to really slow down the propagation of nerve impulses. So then a person's muscles might not work as well if that's where the neuron is ending because they're getting the message so slowly. Okay? So this is at the end of the axon, and it looks very similar to what we've seen at the neuromuscular junction. This is the axon terminal right here. That's the axon terminal. Here's the synaptic cleft. And then here we have another neuron. So we call that the postsynaptic neuron. When we were talking about the neuromuscular junction, that postsynaptic neuron, we were talking about the motor end plate. We weren't talking about the postsynaptic neuron. Okay, so this is another neuron. So the action potential, the nerve impulse, which carries those action potentials, is coming down that axon segment by segment, and it finally gets to the axon terminal. Now we said inside the axon terminal, we have these little vesicles, and these little vesicles are filled with neurotransmitters. And the only neurotransmitter we've been talking about to this point is acetylcholine, or ACH. That's all we've been talking about is acetylcholine. So when that nerve impulse reaches the synaptic terminal, it causes acetylcholine to be released into the synaptic cleft through what process? Exocytosis. Exocytosis releases ACH out into the cleft. So here we have acetylcholine out into the cleft. On the postsynaptic neuron, we have a gated channel over there. It's a sodium gated channel. It is a chemical gated sodium channel. Okay. So there's a little receptor on it for ACH, and so ACH will bind to that receptor. So that's what happens. Okay, but let me back up a minute here because I need to go over this step here. On the axon terminal, we're going to add a step that we didn't talk about with the neuromuscular junction. Okay, on the axon terminal, there are these voltage-gated calcium channels. And it's the calcium channels here, when calcium comes in, this is what's going to cause the exocytosis. The calcium coming in causes the exocytosis of acetylcholine. Right? So the nerve impulse opens up these voltage-gated calcium channels. Calcium is higher outside the cell than it is inside the cell. So that causes calcium to flood into the axon terminal. When calcium floods into the axon terminal, it causes the exocytosis of those acetylcholine molecules and releases acetylcholine out into the synaptic cleft. So that's really the first, the first step is you need the action potentials. The second step is you need these voltage-gated calcium channels to open up, right? The third step then is after those calcium channels, or after calcium has caused the exocytosis of ACH, and ACH travels across the cleft and binds to its receptor, that opens up those sodium-gated channels. We're back to where we started. This was where we were talking about those chemically gated sodium channels on the dendrites and the cell bodies of the axon. It's back to where we started. We're starting at a graded potential again. The gated channels open up, sodium moves in, we get a graded potential. Then if it reaches threshold, we get an action potential. So we can keep passing these action potentials on from neuron to neuron. They can just keep going. There can be where this occurs here, this is called a synapse. Okay? And that can just keep going and going from neuron to neuron. Right? Now, when we were talking about the neuromuscular junction, when in that muscle, if that were a muscle, the muscle contracted after we got an action potential on the muscle side. But if this postsynaptic cell is another neuron, then we're just going to have an action potential occur at the axon hillock, and then it'll be passed down the axon. We have to end this process, though. And by ending the process, we'll then end the lecture. There's another chemical, there's an enzyme inside the synaptic cleft that's called acetylcholinesterase. ACHE. Anything that ends in ASE is an enzyme. Acetylcholinesterase. So it's acetylcholine and then esterase. Acetylcholine esterase. ACHE. Right? So what acetylcholine esterase does is it breaks down acetylcholine. It breaks it apart. It'll take it off of the receptor. It'll take it out of the synaptic cleft. It'll break it down into acetylcholine, into choline and acetate. And the choline is going to be transported back into the synaptic terminal where it can be reused, right, to make more acetylcholine. So, you need to know what happens if you don't have acetylcholinesterase. Well, you're not gonna break down acetylcholine, and then acetylcholine will accumulate inside the synaptic terminal. And then what's that going to do to the cell over here? It's going to cause it to be stimulated. Right? If you have an accumulation of acetylcholine, it just keeps these gates open, keeps them open for longer. All right, are there any questions? So, all right. That will be it for today then.